Our next speaker is Givanna Putri from WEHI. Yeah. And Giovanna is going to tell us about Spectre. Yeah, thank you. Spectre Toolkit for rapid analysis of cytometry data. Yeah. Cool.
Good afternoon, everyone. So my name is Givanna. I'm from the Belinda Fitzsimmons lab at the Bioinformatics division at WEHI. So I'm going to be talking about our Spectre, our package called Spectre, which is basically a toolkit for analyzing and exploration of cytometry data. So there have been quite a couple of cytometry talks today. So this is slightly different in a way that I'm going to start with when I talked about cytometry, I did not mean cell sorting. What cell sorting and flow sorting and cytometry does share similar concept and techniques in that we do stain cells with antibodies. But what you get at the end of a cytometry experiment is basically quantification of up to 50 proteins of millions or hundreds of thousands of cells at the single cell resolution. So as I've mentioned before in my previous Abacus talk, how we traditionally analyze cytometry data is by using manual gating, whereby we iteratively draw polygons and refine them across multitudes of scatter plots, each illustrate an expression of 2 proteins just to isolate the cell type of interest. And as you can see from this animation I just played here, the process is time consuming, it's tedious, it's not scalable, it's extremely subjective. And this is the prime motivation for why we developed Spectre. So this was developed when I was doing my PhD with 2 collaborators, Tom Ashes and Felix at University of Sydney. So very briefly, the Spectre is an R package that contains a bunch of wrapper functions around very carefully curated list of computational tools to analyze cytometry data. So the 3 principles that drive the design of Spectre is simplicity, flexibility, and usability. Simplicity in that we want our package to be easy to use, especially for those with extremely limited programming experience. Flexibility in that we don't want to impose any ordering in which function you need to run first. So the whole idea with this is that do what you want in your analysis strategy as long as it makes sense, and just take whatever you need from our package. And lastly, usability is also tightly linked to the flexibility in that we would like anyone to be able to just plug any single function within the package and throw it straight into your existing workflow with very, very minimal data wrangling. So we were able to achieve this by basically storing and managing our data using the data table package, which is basically just an enhanced version of the native R data frame. And to assist with the interoperability with the bioconductor packages, we have provided functions to actually convert your data table into single cell experiment or the flow called flow frame object, which is quite commonly used for cytometry data. So a data analysis using Spectre may look something like this. So take your cytometry data, which you can present as a table. But it's slightly different from single cell in a way that we typically represent the cells as the rows because we tend to have much more cells than proteins and the proteins as the columns. What we typically do then using Spectre is we first do arc-sign transformation. And after that, you can do batch alignment if you have data from different batches. You can run clustering. So in here, we have FlowSum and Phenograph, which are the 2 quite popular algorithms, as Elijah has mentioned. And then slightly different from single cell, we do subsampling first because we have way too many cells we have to subsample before we run UMAP. because otherwise it would just take way too long. And then after that, you can use Spectre to draw UMAP plots to visualize any protein expressions that is of your interest, and then use that information together with the clustering information to do cluster annotation. And then typically what you can do with Spectre after this step is generate what we call summary data, whereby you calculate, say, proportion of cells captured within a given cell type or a mean expression of any given protein expression for a given cell type. And then we provide functions then also to visualize it as a heat map or to draw, visualize it as a violin plot with very, very simple, I have to stress this, very simple statistical tests just to compare those features across different biological groups. So I will quickly just show you the kind of result that we can get by analyzing data using Spectre. So this data set is a West Nile virus data set. It's a mosquito-borne virus. So the data sets we contain two different time points. One is called pre-infection, whereby it's a mice brain sample that we take before any infection of any virus. And then the second one is called post-infection, where we take the samples from the mice brain 7 days after it's infected with the virus. So take this is SEO data. So again, we do oxide transformation, we run flow sum to cluster data, and then we subsample, run UMAP, do cluster annotation, and we resolve 5 different cell types, as you can see here. And then when we actually compare the pre-infection and 7 days post-infection, just from the UMAP alone, we can actually see that there hasn't been that much change in the resident microglia population, but a completely different story for the infiltrating immune cells. But we see a sort of massive increase in their numbers 7 days post-infection. And then we do an extremely simple differential abundance test where we simply do a Cross-Wallis and Wilcoxon-Wren test to compare the number of cells in each of the cell types. And what I'm showing here is just for these 2 different cell types, microglia and infiltrating macrophages. And as you can see from the actual test alone, the p-value is not significant for the microglia. So we were right, the microglia hasn't changed much, but the infiltrating macrophages, it just suddenly exploded, which actually makes sense from the biology. And then after that, we do a very, very simple differential expression test where we basically just measure the Z-score for the mean expression of 2 proteins, CD11B and Ly6C, for each of the cell type we have uncovered, comparing the pre and post-infection. And we illustrate that as the heat map here I'm showing at the bottom. Sorry, it looks a little bit small. But the key take-home message here is that using the heat map, we can clearly see that the expression of those 2 markers or proteins, CD11B and Ly6C, are significantly lower pre-infection compared to post-infection. So as I've previously alluded before, within the package itself, we have ability to do batch alignment. So this work is predominantly driven by Tom Asher's. So we consider 2 different batch effect, minor batch effect and major batch effect. Minor batch effect is the case whereby if you have cells from different batches, when you try to cluster them, you can still sort of largely capture the cells from different batches, but of the same cell type in the same cluster. So to handle this sort of minor batch effect, we can employ a fine alignment strategy whereby we basically just run Cytometry algorithm. So briefly, very briefly, how Cytometry works is that it basically takes your healthy samples and then it runs flow sum clustering and to compute a quantile-based conversion, which it then used to convert those healthy samples first. And what happened with the disease sample is that it takes the clustering result from the healthy samples and map them onto the disease samples, and then use the quantile-based conversion it previously calculated to convert them. And you end up with an aligned data. So that's great. But what if you have a major batch effect? What if when you run clustering, your clusters, each of your clusters basically captures cells from exactly just one batch? You can imagine Cytometry will not work because it works on the premise that you can capture cells from different batches within the same cluster. So Tom came up with this strategy whereby you first apply what we call a coarse alignment, whereby you basically treat all the cells in your healthy sample as if it just belonged to one giant cluster. And then you just compute the quantile-based conversion and do the conversion. And you do similarly for the disease sample as well. what you get is partially aligned data, which you then can pass on to the final alignment strategy, which I just previously described, to get a better aligned data overall. So an extension to the batch alignment algorithm is the feature to do a cell type label transfer. So this is typically very handy if you have analyzed data from one batch and you've annotated them, did all the hard yards. But then you have data from a different batch and you don't really want to repeat your experiment again. The analysis again. So what you can do is basically employ one of the batch alignment strategy and then just train a k-nearest neighbor algorithm on those one of the batch that you've annotated before and just use it to transfer the annotation. So all the features I've just mentioned are already been published. So if you want to know more, please download the paper and have a read. And all the code, everything is all available on GitHub, and you can start using Spectre by just using the remote function, remote package, and just install it on ARM. So the next few slides I'm going to talk about are basically features that we're still currently actively working on, and we would like to release as part of version 2 of Spectre sometime next year. So the first feature that we'd like to do is basically having the ability to integrate data across different cytometry technology, and ultimately as well to integrate CytSeq and cytometry data. So this will be something that me and my lab head Belinda Phipson will be working on. So the second big piece that we would like to also release as part of version 2 is adding support for spatial data analysis, starting with IMC data, which Alice and Eliza have described. But briefly, just very, very briefly, IMC is a subcellular spatial technology that's based on mass cytometry, whereby you have a tissue section stained with antibodies, blast them literally with a UV laser, and then quantify the material using mass cytometry. And I'm sure you have seen enough IMC image for today. So just very briefly, what we would like to do is basically have the ability to import your segmented image, segment them however you like, using whatever protocol is your favorite, import them as a data table, and then analyze them as if they are your suspension-based cytometry data. And also we like to add some features to calculate spatial features like cell densities, distance between cell and neighboring cells. And that's pretty much it. And the oldest code for the spatial analysis are all already also available on GitHub for anyone to try. All you need to do is just download and install the development branch of Spectre, and we'll appreciate and we'll take any feedback on how to improve all these steps. So please do give them a try. And also, if you like to try out Spectre, in our website here, we have a bunch of protocols and step-by-step workflow on how to analyze your data using Spectre. And thanks for listening. I would like to acknowledge all the people in here, especially Tom and Felix from the Immune Dynamics team at the University of Sydney. They are the two other co-developers for Spectre alongside myself. You can scan this QR code if you would like to know more about the team. And of course, I can't thank Belinda Phipson from WEHI, my lab head, enough for her continuous support for the project, and the bioconductor for providing the scholarship for me to attend BioC Asia conference. Thank you.
Thank you, Giovanna. There are questions? Yeah, there's one from Marie here.
Yeah, very nice talk. I was a bit curious, how did you come up with the idea of Spectra? I mean, comparing, so there are a few works like the Catalyst package, the DeepSeed from Bioconductor, both of them also provide some methods for like differential analysis. So did you, yeah, I'm a bit curious.
Yeah, so Spectre is all the credits actually to Tom. So this was what he started off as his PhD project. He just basically needed something that is easy for him to use to analyze his own data set. And he found it irritating having to interrogate a lot of these objects like flow frame objects, importing his data and so on. And he found it is a bit difficult to interoperate between different packages as well, because they seem to like different objects. So then he came up with this concept of Spectre. We need to build something such as it's easy for someone that is not very sort of experienced with programming, where what he was like when he was doing his PhD, to actually analyze the data using R rather than just using FlowJo software. So this is the big motivation for Specter. And then as I was doing my PhD, he became my supervisor. And he said, do you want to work on this? And I said, of course, why not?
Thank you.